Fighting Blindness Canada's Viewpoint is an education series that brings you the latest in vision research presented by health experts from across Canada. The session you're about to watch is a recording. To learn more about the research we fund and upcoming events, please visit our website at fightingblindness.ca. This Viewpoint is proudly presented by Bayer and supported by Abbvie, Apellis, Astellas, Biogen, Janssen, Novartis and Roche. Thank you for watching. We hope you enjoy this session and share your thoughts with us in the comment section below.
Thank you for the kind introduction. I'm very happy to be here. So today I'm going to be talking about inherited retinal degenerations. So these are conditions that are inherited due to a genetic mutation. And I'll be talking about the different interventions, that we are working on to try to address that disease process. So I just wanted to say that even though I'm focusing on inherited retinal degenerations, I know the people that are here today have various, issues, not just IRDs, inherited retinal degenerations, but also myopic degeneration, birdshot, different types of conditions. So I'll try my best to kind of speak more generally when I can, but I'm going to be coming from a perspective of inherited retinal degenerations. Just use that. Or maybe I can just put it here. Is that okay? Okay, let's try that again. Okay, so these are my disclosures. I do consult and do work with a lot of sponsors in order to do these clinical trials. And I just want to start with just a general overview of inherited retinal degenerations. These are rare diseases, but they're entirely heterogeneous, meaning that there are many different types of degeneration caused by many different genes. And most of what happens is that usually there is a genetic mutation, a mutation in a gene that's vital, for retinal function, either for the photoreceptor, which is the light-sensitive nerve in the retina, or the retinal pigment epithelial layer, RPE, and that's one of the supported layers. And a mutation in a gene that's important in those cells eventually leads to degeneration of either the photoreceptors or the RPE cells. And then that in turn leads to subsequent degeneration of other layers of the retina, the choroidal layer, for example, and even remodeling changes that occur in the rest of the retina. And as that goes on, it also affects the brain. With loss of vision can also affect remodeling in the visual part of the brain. And so for a long time, there really was no treatment for these conditions. But over the last 10, 15 years, we've come a long way in developing different avenues, different strategies. And that can roughly be broken down into gene therapy, cell-based therapy, and small molecule. So for gene therapy, I'm sure you've heard that term a lot. Typically that means that we're trying to repair the gene that has a mutation. There's different ways to do it. We can replace it with a good copy of the gene. We can do gene editing to try to fix the very mutation in the patient's DNA. There's also RNA editing, right? So RNA is an intermediate step between DNA, what's in your DNA, and what the cells in your body's making, protein. And so we can try to fix the intermediate step. But in all, what we're trying to do is get the retinal cells to start producing normal protein. And in doing so, allow the cells to function more normally. And hopefully that translates to slowing down or stopping the degeneration. There's also another special type of gene therapy. It's called optogenetics. Now, in this case, we're not actually trying to fix the genetic mutation. This is usually employed in patients who have pretty advanced disease and they've pretty much lost their photoreceptors. And so what we're trying to do is we're trying to transform some of the remaining nerves in the eye that normally doesn't sense light. We turn them into photoreceptors, or at least quasi-photoreceptor-like nerves. And that brings back some vision, to an extent. We call that artificial vision. So it's a form of kind of rehabilitation. And the other things you can do with gene therapy, is also something called the genetic biofactory approach. So there's knowledge that there are certain proteins, growth factors, things that can hopefully slow down degeneration. And you can use gene therapy to actually hijack a part of the retina to start producing some of these proteins and hopefully, again, slow down degeneration. Now for the category of cell-based, right? I think stem cells, everyone's heard of stem cells. Well, there's different ways to use stem cells. One is, again, for neuroprotection, and that means these are cells that are producing specific proteins and growth factors, and these are good for the nerves that are degenerating, and hope that these slow down the process of degeneration, and you would inject these cells into the eye. But then the other way, which probably what most people also think about, is being able to repair, right? Being able to regrow the retina that's been lost. Okay, so that's the holy grail of medicine, right? To be able to actually regrow what has already degenerated. And that is as hard as it sounds. And I'll talk a little bit more about that later. So then in a whole nother category, we call it small molecules. So these are drugs, medications, pills, that you could take to hopefully slow down degeneration. And many of these medications either are an antioxidant or anti-inflammatory to kind of mute some of that degeneration process, or it can actually inhibit a specific pathway, that we know about in the disease process. The other thing I want to cover is, well, how does that all these strategies apply, to me as a patient? Well, we can break this down into the disease course, right? So interventions that slow or stop the degeneration work best, right, when applied early on in the disease course. So kind of early to mid-phase disease when there's still some intact vision and there's still retina, to be saved. And so that's gene therapy, right, RNA therapy, and also small molecules that slow down degeneration. So that's the way we think about it. Whereas disease that's more in late phase, advanced phase, where there's not as much retina left, to save, that's where we start thinking about artificial vision, right? Optogenetics, as I talked about, and also trying to regrow or replace the retina with cell-based transplants. Okay, so now I'm going to speak a little more specifically about each of these categories. And the first one I'm going to talk about is photoreceptor replacement, right? And that's a hot topic because in this case, right, it doesn't really matter how you ended up with retinal degeneration, how you lost your retina. right? Because if we can develop a way to replace that and regrow the retina, right, it doesn't matter if it's retinitis pigmentosa or age-related macular degeneration. And so one of the ways that have been developed and used by some of the companies who have done clinical trials recently is taking these cells from fetal donors. Now, there's a kind of an ethical issue there, for, having fetal donors. But what they did is they had these donors and they isolated the retina, right? Fetal retina, and they were able to purify what is called retinal progenitor cells. These are cells that are going to become, retina and photoreceptors. And they process that into a treatment, into a cell suspension that you can inject under the retina. And the idea is that you inject these cells and they would hopefully completely differentiate into photoreceptors and then repopulate the area where there's degeneration and then reintegrate and hopefully provide vision, okay? So one way to get around the ethical question, and there's at least one company doing this, is to have adult donors, right? So you can actually take adult tissue, and that can be, white blood cells. You won't miss some blood drawn from your arm. And you can differentiate that or de-differentiate them, basically, induce them to become pluripotent stem cells, iPSCs. And then from there, you can then differentiate them into photoreceptors. Okay, and so there's one company, Blue Rock Therapeutics, is using this very method, right? And so there, it's not so much of an ethical question. And again, we're hoping to inject those cells under the retina, right? And hope that those cells repopulate the degenerated area and provide, vision. Now, there's also one more company called Endogena. And they developed a proprietary small molecule that they think can trigger endogenous stem cells. Endogenous means that the cells are already there, right? So we're not putting new cells into the eye. So there's a part of the eye called the ciliary body where there's already cells there, stem cells. And there's this molecule that they believe can stimulate these cells to differentiate into photoreceptors and migrate into the retina. So that's currently an ongoing clinical trial, which, if it works, would be amazing. And so, in summary, there's been 3 clinical trials in the inherited retinal degeneration space for these sort of cell-based treatments in the last 10 years. They haven't all been successful, but each time, we do one of these, we learn a lot. They aren't moving on to advanced phase clinical trials, but at the same time, there are other companies, that are coming on and doing new phase 1 clinical trials. For example, Blue Rock Therapeutics, like I, went over, and then also another company called Astellas is developing, hopefully the next generation, right, cell-based treatments. One thing I do want to point out is, why is that it's as hard as it sounds, or what are the challenges? Well, one of the studies, the phase 1 study that was done by Third Military Medical University in China, and they published on this, they showed that there was an issue with those cells staying where they're supposed to. We're really good at taking pictures of the retina, right? And so we can see, when you inject cells under the retina, you can see stuff there. But unfortunately, in that study, as they monitored those patients over time, those cells disappeared. And so it's probably due to some sort of rejection. These cells are not really part of your body. And much like organ transplant, we think that there's probably an issue with rejection. Even though we typically think about the eye as being immune privileged, meaning that you can put stuff there that's not your cells and it'd be okay, but I think it's going to be a little harder than we thought. Okay, so, but I think there's still a lot of companies working to hopefully bypass that, either with different kinds of cells or using different regimens for immunosuppression. Okay, so the next thing I'm going to talk about is neuroprotection. So again, I said that neuroprotection is basically a drug or a pill that you would take to hopefully slow things down. There's lots of interest and clinical trials around Stargardt disease due to mutations in the gene called ABCA4. A lot of these medications are designed to inhibit the disease process. There's also another big one that started for retinitis pigmentosa called N-acetylcysteine. So it's a compound that we think is an antioxidant, and it can hopefully slow down the degeneration no matter what kind of retinitis pigmentosa that you have, and that's currently in phase 3 clinical trials. I won't have time to go into all the different ones, but there is one interesting one that I do want to talk about that I touched on earlier, about how you can inject cells into the eye that produce a protein that can offer protection and slow down degeneration. So that's a company called J-Site, right? So they've developed these stem cells that they believe secrete growth factors and proteins that are good for the nerves and reduce the amount of degeneration. And so they finished their phase 2 clinical trials, and the results are quite compelling, right? They found that a portion of their patients, about a quarter of the patients who received the highest dose of cells, they had an improvement of 15 letters on the eye chart. So that's 3 lines on the eye chart. And so that's quite amazing. And so they're now going into phase 3 clinical trials to make sure that it's repeatable, right? And when you do phase 3 clinical trials, that's kind of considered the pivotal. That's the data that you need to try to convince the FDA and everybody else, right, that it's working and that it ought to be approved. Okay, so we're very excited. It's going into phase 3 clinical trials soon. Okay, now finally, gene therapy, right? This is probably the biggest topic of the 3. And remember, gene therapy, I said, is an attempt to fix the gene at the level of DNA or RNA in order to normalize protein expression in your retinal cells and thereby slow or stop the degeneration process. And so I had mentioned earlier that there's hundreds of genes that cause retinal degenerations, right? We don't have a clinical trial program for every single one of them, but there are quite a few. And the amount of activity over the last 10 years has exploded. And so right now, we have multiple clinical trial programs for about a dozen, of these genes. And sometimes for one of the genes, there's 4 or 5, different clinical trials, different companies, that are trying to develop their own version of gene therapy for that gene. Overall, there's roughly 3 kinds of gene therapy strategies. One is called gene augmentation. That's where we just deliver a good copy of that gene, to the retina. Another one is called gene editing, and that's where we use a specific type of gene editing material to go in and actually fix the mutation in your DNA, okay? And another one is called RNA editing. So really just want to tell you there's 3 types. I won't have time to go into all the details there. And there's, pros and cons to each of those strategies. But there are dozens and dozens now of clinical trials, and we're able to be, part of most of these clinical trials at KCI Institute. And again, I'm not going to have time to go into all of them, but I do want to talk about 2 specific gene therapies. One, I'm going to talk about Luxturna, which is the first FDA-approved gene therapy. And then I'll also kind of talk about, give you an update on one of the clinical trials that we're doing for another type. So the first FDA-approved gene therapy for the retina is called Luxturna. It was approved in 2017. And it was created to treat retinal degeneration due to a mutation in a gene called RPE65. RPE65 causes retinitis pigmentosa and Leber congenital amaurosis. And I do want to take a moment here to say how we got to that point, right? It didn't just magically happen overnight to have this approved product. The whole process of going from phase 1, phase 2, phase 3 clinical trials took about at least 10 plus years. And in the beginning, there were multiple companies developing treatments, and not all of them made it to phase 3, right? And so Spark Therapeutic, they were the ones that made it to phase 3 clinical trials, and they were able to show everybody and the FDA that this product, really was working. And so I want to show you a video. This is the kind of benefit, the kind of data that they showed to the FDA to prove that the product works. So the thing that they showed in the study was that patients were able to navigate under dim lighting after treatment. Because in this condition, night vision is really poor. Okay, so on one side of the screen, I'm going to get the video going. On one side of the screen is one of the patients trying to navigate this maze before treatment. And on the right side of the screen is after treatment. So what the videos are showing is before treatment, and this is under dim lighting, the patient could not even start the maze. The patient could not even see well enough to even begin. So, that was basically kind of failed, could not do that test. After treatment, the patient briskly walks through the maze, almost like normal. Okay, so it was a huge effect, to have this gene therapy. And so that's the kind of data that went to the FDA, and it was fast-tracked, and it was approved. And now I want to talk about one of my patients. Okay, so this patient was diagnosed in 2017, the same year that Luxturna was approved, and he was 4 years old at the time. And so we very quickly turned around and treated him with Luxturna. And treatment does require surgery, so the patient underwent anesthesia, had both eyes operated on. But before treatment, he needed a headlamp to see anything indoors. Otherwise, it was just not bright enough. He needed a cane to navigate. That was his life. And so in the surgery, we carefully injected the gene therapy under the retina, and we can use imaging systems during the operation to make sure that we put it exactly in the right place. And this is what happened. So in 2017 to 2018, just before he had the treatment, his vision was getting worse year by year. His vision was 20/50 in 2017, and in 2018, it was 20/80 in the right eye and 2100 in the left eye. So very rapidly getting worse. And then we treated the patient in 2018, and after treatment, he he was 20/40 in the right eye and 20/30 in the left. And he's been pretty much the same since then. Not only that, we also were able to get fancy images of what the retina looked like. And in degenerations, we can see the layer of photoreceptors, start disappearing. And after treatment, right, and we've never seen this before, before Luxturna was approved, we started seeing the retina, some of it started coming back. Not that we're regrowing the retina, but that the existing retina was being rehabilitated. Right, and so quite amazing. Visual acuity was getting better, the pictures were getting better, and even the ERG was getting better. The electroretinogram, some of you may have had that done on you, which shows us that electrically the retina was functioning better than before. And so fast forward, he was able to give up his headlamp and give up his cane. He didn't need that anymore. And it was actually kind of hard to ask him to give it up. It was part of his identity. That's all he's ever known. But he was finally able to go to the aquarium and enjoy the aquarium and just be a kid again. And Canada is following suit, right? It was Health Canada approved Luxturna in 2020, and I think the different provinces are starting to cover it. I think I read that Ontario, right? It's all the provinces now, but I guess Ontario was kind of made the news in 2023. Very exciting to see that basically spread throughout the world. And so right now we have many different clinical trials going on. We are a commercial treatment center for Luxturna. We have clinical trials for X-linked retinitis pigmentosa due to RPGR. We also have clinical trials for other types of retinitis pigmentosa due to PRPF31, rhodopsin, PDE6A, PDE6B, and we also have multiple clinical trials for Stargardt disease due to ABCA4. Now, one of them I do want to talk about more specifically. One of them is a phase 1/2 study for another type of Leber congenital amaurosis. And this is a condition that's due to a mutation in a gene called GUCY2D. We call it Gucci2D. So this type of Leber congenital amaurosis is among the most severe types. And this is also done with a surgery, and we inject the gene therapy carefully under the retina. Okay, and so far it seems to be well tolerated. Okay, this is a phase 1/2 study, which means the primary goal is safety, but we also look to see whether or not it works. Okay, and one of the tests that we did is something called FST. FST is a test where we determine what the patient's light sensitivity threshold is. How sensitive is their eyes to different levels of light. And we can test it with white light, blue light, red light. And just 1 month after treatment, the eye that got the gene therapy was immediately much more sensitive, no matter what color light we checked. And that we observed on average for a lot of the patients in the highest dose, gene therapy cohorts. Okay, and I want to talk specifically about one of my patients. So she's a teenager, right? She grew up with very poor vision, right, with this condition. And we did the gene therapy, and like all the other patients, right, her light sensitivity improved in that eye. But she was kind of shy, and she didn't really, say a whole lot at these visits. And then that winter, her mother captured a video, and we don't have audio here, but I'll tell you what's going on. So here's a video of what the mother sent us. And it was basically her reaction to seeing snowflakes for the first time in her life. So it was really heartwarming. We shared that with the team. We checked with the patient and her mother. We checked with the sponsor, the company, and everyone said it was okay to talk about it and release it. So it's on the internet because it's really heartwarming, right, to see the hope, right, to see the hope that even phase 1/2 trials can bring, and the impact, right, it can have on patients and their families. And also to raise awareness, right, to raise awareness about this condition and all the other conditions, right? So really wanted to share that with you guys today to hopefully give you some hope, with whatever condition it is that you're dealing with. And so, in summary, I want to say that there's a lot of these interventional clinical trials right now. It can be hard to stay updated or know what's going on, and that's why we're having a day like today, and I can introduce some of that to you, give you some of the details, and give you a chance to ask questions. But it continues to grow rapidly. There's a lot of interest in these conditions and to develop novel treatments. Right? I've talked about gene-based treatments, cell-based treatments, small molecules, right? And all that is using different strategies to try to slow things down, right? Stop the degeneration and hopefully replace what's lost. Luxturna, right, for RP65 related degenerations is still our kind of one approved treatment, but I hope it's not the last, right? We have lots of research going on, and the data, is compelling, and I'm hoping there's going to be more FDA-approved treatments in the future. Thank you.
Thank you so much, Dr. Yang. That was great. And I would like to say our next session is our Ask the Expert session, so we will have lots of time for questions then. But why don't we take, we have a few minutes, we could take a couple questions now. If there's anyone in the room who has a question, we can bring the mic to you. Okay.
Thank you so much for your talk, Doctor. My question is just out of curiosity, what made you decide to go into studying this field?
Well, I was asked that question earlier today, and so I think I've always, wanted to do things that not everybody else was doing. And when I was in training, as a medical student, I felt like, well, things like cancer and heart disease, there's lots of people doing that. But then I started seeing really rare things, rare conditions that sparked my interest, and things where there's just not enough information. There's no treatment available. And I think that really inspired me to study rare diseases and develop new treatments, for patients who've not previously had really any options. Fast forward, I also got interested in the eye and decided that that's what I want to do for my career. And so I kind of put those two together and became a specialist, for inherited retinal degenerations.
Hi, my name is Gabrielle. Actually, I guess it's more just like, I don't know, I'll be brief if I can, but I guess I just wanted to say that I don't have a retinal condition. I have a corneal condition. It's Peters anomaly and aniridia, and they're glaucoma related. And I just kind of wanted to let people know that it is kind of, yeah, the gene therapy is possible kind of thing from the retina, because even with corneal, recently, a month ago, I got a limbal stem cell transplant. So that wasn't something possible even 5-10 years ago for me, it's still like a new procedure. So yeah, I could probably like talk more later in the next session, but I just wanted to bring that up.
Great, thank you for mentioning that. Yeah. Yeah, and I think that's another good example where, I focused on a very specific slice, of rare diseases and, certain conditions, but I think, the patients here represent a much, wider, spectrum of disease, and thanks for sharing.
We have a bunch of questions online, so let's take one from our live stream audience as well. I can pull it up here. So there was a question from Jerry, and Jerry was asking if you could explain more what you mean by the brain remodeling in the visual cortex.
Yeah, sure. So studies have been done using MRI, which is the way to image the brain, and you can actually image the brain in a way that tells you how the brain is working, okay? It lights up differently under MRI. And so for patients who've had vision loss, they found that those parts of the brain that normally process vision, they start doing other things. They start processing sound or touch, once enough time has passed. And so these are signs that those parts of the brain, when they stop receiving visual input, they'll start doing other things. They get recruited, to do other things. And so I bring that up because we just have to be conscious, of the entire degeneration process, when we're thinking about, treatments.
Oh, there's one at the back over here. Oh, yeah.
Hi, Dr. Yang. I have RP, and I've had a couple rounds of genetic testing. With genetic testing, obviously, there's, as you said, 100 mutations, and they haven't been able to link my mutation. So gene therapy is not an option for me. But I'm very fortunate to see Dr. Gregory Evans, as I think many of you know. And he's given me hope, and I hang on to that hope that within my lifetime, and I ask them every time I see him, will there be a treatment? And he says yes, that stem cells are my hope. And I'm not doubting Dr. Gregory Evans, but I would also like your professional opinion on that.
Good question. I don't doubt him as well. We're colleagues. And so you're not alone, first of all. So we estimate probably 25% of patients with RP don't get back a positive result on their genetic testing. Doesn't mean it's not RP, it just means we don't know all the genes yet. Okay, so we're still building our database. We don't know when it is we're gonna find yours, right? But, we're working on it. And there's hope. Even if you don't have your gene, I was mentioning, there's different strategies beyond gene therapy, right? So N-acetylcysteine, for example, that's one of the small molecules that we hope can slow down degeneration. It's a pill that you would take. It's in phase 3 clinical trials. And so for that, it doesn't really matter what kind, what genetic your type of RP is. And so hopefully, if that gets approved, that's something that could help you slow down your degeneration. And he's right, stem cell therapy, again, it doesn't matter what kind of RP you have, but once you've lost your photoreceptors, hopefully in the future, we have the ability to kind of repopulate that part of your retina with stem cells so that we can regrow your photoreceptors and rehabilitate vision. And then finally, also optogenetics, I kind of talked about at the beginning of the talk. It's a way to give back some artificial vision. And so even though after you've already lost your photoreceptors, right, you still have a lot of remaining nerves left. They're just not photoreceptors. They're not light sensitive. So optogenetics, we basically engineer using gene therapy to convert some of your remaining nerves that aren't normally light sensitive to become light sensitive, and thereby giving you back, some amount of vision. Okay, so there's definitely a lot of other strategies, that could be, helpful for patients where you don't know what gene you are.